Hi everyone and welcome to the presentation of our poster, Transfer Learning for T-cell Response Prediction. This is work by me, Josua Stadelmaier, and my supervisors, Brendan Malone and Ralf Eggeling. In this work, we are studying the machine learning-based prediction of T-cell response for peptides that are already presented on MHC molecules. This prediction task is challenging due to the heterogeneity of T-cell response datasets in the form of a multi-domain structure. Additionally, the amount of available data is relatively small. One challenge arising from the data heterogeneity is the risk of shortcut learning. With shortcut learning, we mean that a model picks up simple patterns that are not biologically meaningful. Our contributions are a careful, domain-aware evaluation that accounts for possible shortcuts. We investigate a transfer learning technique called adversarial domain adaptation to reduce shortcut learning. Based on the results of our experiments, we propose using a transfer learning approach which entails fine-tuning a pre-trained model for individual peptide sources. We then demonstrate that this improves the accuracy of T-cell response predictions. We have compiled a dataset from the immune epitope database. Each data point consists of the peptide sequence, the binary label indicating whether T cells have responded to the peptide with interferon gamma release, the allele of the MHC molecule presenting the peptide, and the peptide source, that is, the organism or the virus the peptide originates from. Our dataset covers both MHC class 1 and MHC class 2 peptides, and overall we have about 15,000 positives and 21,000 negatives. We conduct our experiments using a Bayes model, which we call Bayes transformer. The gray boxes show the components of this model. We start with just a peptide sequence as input, which gets embedded into a sequence of vectors. A single-layer transformer encoder then updates these vector representations by letting them interact with each other. After that, the sequence of vectors is combined into one vector, the latent peptide representation, using mean pooling. Based on this vector, a simple multi-layer perceptron then predicts the T-cell response labels. The boxes with the red background show the components of our adversarial domain adaptation approach, called ADA transformer. Here, the latent peptide representation is fed into another multi-layer perceptron, called adversary, which is trained to predict the source of the peptide. The ADA transformer is trained with the objective L-ADA, which not only includes the minimization of the T-cell response loss, but also the maximization of the adversary loss. This encourages peptide source independent T-cell response predictions. Before I show you the results of the adversarial domain adaptation approach, I briefly explain our domain-aware evaluation. The data features two domain structures, the MHC alleles and the peptide sources, with varying label frequencies between peptide sources and between MHC alleles. This leads to the risk that a model can pick up the domain identities and use them as a shortcut for predicting T-cell responses. Our domain-aware evaluation ensures that performance estimates are not inflated by such shortcuts by first computing shortcut-free ROC-AUC scores on data subsets containing only one MHC allele and one peptide source, and then combining the subset results into one AUC score. Looking at the cross-validation results, the AUC scores of the Bayes transformer are highest if we are not adjusting for shortcuts. When applying our domain-aware evaluation to MHC alleles or to peptide sources separately, the performance is reduced. And when adjusting for both shortcuts at the same time, the AUC values are even lower. This indicates that both shortcuts are actually used by the Bayes transformer. When comparing this to the ADA transformer, which encourages peptide source invariant predictions, we see that the results without adjustments and the results that are adjusted for peptide source shortcuts are identical, demonstrating that the ADA transformer reduces shortcut learning. These histograms of prediction scores further confirm that shortcuts based on the majority label within a peptide source are used. For example, here we can see that peptides corresponding to the blue bars originate from a source that has mainly positive labels, leading to incorrectly high prediction scores for this minority of negatives in this peptide source. The ADA transformer counteracts this effect. For example, here we cannot see a difference anymore between the peptides having the minority or the majority label of their source. One thing that we can also see when looking at the evaluation with both shortcut adjustments is that the ADA transformer does not improve the overall performance compared to the base transformer. To investigate why the performance does not improve by encouraging peptide source invariance, we train per source models and find that there's negative transfer between peptide sources. This motivates our second transfer learning approach, the fine transformer. In this approach, we first pre-train the base transformer on all peptide sources and then fine-tune it separately for each peptide source. The per-source fine-tuning ensures that there is no negative transfer between sources, and the pre-training step enables positive transfer between sources. This figure shows that the fine transformer leads to improved performance over a wide range of peptide sources compared to the base transformer. We also verified with an ablation study that both pre-training and fine-tuning contribute to the improved predictions. Finally, we also evaluate the performance of the fine transformer on a 20% holdout test set. For a comparison with existing models and with personalized cancer vaccines as potential application in mind, we show the results for human peptides only. Since the existing models have been trained on different datasets, we also include a bag of amino acids baseline that is trained on the same data as the fine transformer. As you can see here, the fine transformer outperforms existing approaches and the bag of amino acids baseline for both MHC class 1 and MHC class 2. If you have any questions or feedback, and if you're interested, feel free to just write me an email or follow me on Twitter or on GitHub to get notified when the paper is out. Thanks for watching.